Hi, everyone. It's an absolute pleasure to be able to participate and contribute to the Becker Education and Engagement Day. It's time to get real about Becker muscular dystrophy. I'll be talking about imaging muscles in individuals diagnosed with Becker muscular dystrophy. The ImagingNMD study is being performed at two different locations, the University of Florida, which is where I'm located, and our partner institution, Oregon Health Science University in Portland on the West Coast. The ImagingNMD study is a natural history study, not a clinical trial. It's funded by NIH and was started in 2010, originally focused on young ambulatory boys with Duchenne. Thanks to the tremendous efforts of many families that were willing to travel to one of our sites, the study has transformed the field. The MR measures that were developed and tested in the ImagingNMD study are helping to inform numerous clinical trials. Our goal is now to leverage all of that valuable data and knowledge that was gained over the last decade to benefit individuals living with Becker's muscular dystrophy. The ImagingNMD study in Becker's has really three goals. The first one is to enhance our understanding of disease progression across different mutations and different muscles. Two, to develop sensitive measures that can be used in clinical trials and quickly and objectively determine whether a potential new drug is going to be effective. And thirdly, to help us understand the relationship between dystrophin levels and disease progression. Our goal is to accelerate therapeutic development in Becker's muscular dystrophy, not for one particular therapeutic target or drug, but really for the entire community. MRI is not able to measure or even detect dystrophin, but it is sensitive to a number of key pathophysiological processes that are important features of disease progression when we lack dystrophin. MRI can detect inflammation and damage. We can measure muscle wasting, and we can measure the infiltration of muscle by fat. And the latter turns out to be a really important feature of disease progression in Becker's muscular dystrophy, as well as other dystrophinopathies. So ultimately, dystrophinopathies can be characterized as being muscle wasting disorders in which there is progressive replacement of muscle by fat. So here is an example. On the left side here, I have displayed an image that is taken of the upper leg of a healthy control. So here we have the quadriceps and here we have the hamstrings muscles. And so typically in a healthy muscle, you can see that the signal is kind of gray and it's pretty homogeneous. In contrast, when you look at these same exact images that we're taking in individuals with Duchenne or Becker's muscular dystrophy, or even symptomatic female carriers, there's one thing that they have in common. And what you can see is that there's a lot more structure in the muscles. And then importantly, this bright or this white signal is really the consequence of the fact that the muscles are being infiltrated by fat. We can quantify this infiltration using an MR imaging approach. So in this case, we take water images as shown on top, we take a fat image, and if we combine these images just right, then we get quantitative fat fraction maps. So in this case here now, we have each of the pixels in this image is now a quantitative measurement of fat fraction, which is defined as fat over water plus fat. We can also visualize it as shown on the right side here by using these images. So here now we can see that fat is displayed in green and red is water, which is really primarily muscle. And so you can appreciate the infiltration of the fat inside of the muscles here. And you can also see the variability that exists across the different muscles, with some of the muscles being more preserved than other muscles. We can also use this methodology to carefully track disease progression, or in case of an effective treatment intervention, potentially the stabilization or even the reverse. For example, here, just as a proof of principle, these are images taken of the lower leg of an individual with Duchenne. Here, this young boy is 7.5 years old. And if you look at the calf muscles, they look really well preserved. There's not a lot of green, not a lot of fat inside of these muscles. But if we come back a couple of years later, you can see more and more fat inside of the muscles. We have done these measurements now in well over 200 boys with Duchenne and carefully characterized the trajectory of fat infiltration across different subjects. So here is on the graph here on the right shows you fat fraction measured in one of the quadriceps muscles as a function of age. And each of these dots represents a measurement performed in one of the participants. And there's two observations that I would like you to focus on. One, you could see the overall trajectory. It almost follows an S shape with slow rates here early on, and then it kind of pivots and you have this rapid increase until it levels off again, simply because the muscle is replaced by fat. The second point is that there is a lot of variability across subjects. For example, here you have an individual in which you can see very rapid fat infiltration and other individuals that have a more modest disease progression. I should also point out that even though all of the individuals that were originally enrolled were categorized as Duchenne muscular dystrophy, we found that the individuals here at the bottom that have this slow rate were re-diagnosed with Becker's muscular dystrophy or an intermediate phenotype. What's not shown here is that the rates, this S shape is true for all of the muscles that we've studied, but the rates are quite different. So at this point, we really understand the trajectory very well in individuals with Duchenne, and we have modeled and characterized it very carefully. In Becker's participants, we are taking an even more comprehensive approach. In addition to these localized measurements, we are performing whole body imaging. And so we're essentially getting images now of the shoulders, the arm, the trunk, the pelvic girdle, as well as the upper and lower leg muscles. At this point, we have collected data in 52 men between the ages of 18 and 62 years old with a variety of mutations. And so what we found so far is that there indeed is significant fat infiltration. However, there's a lot of variability between the different participants. And so here, for example, I'm going to show you fat infiltration in 6 men that are still ambulatory. These are all images taken of the arm, so at the same level. So here in this individual, this is a 21-year-old. And when you look at the biceps and triceps, it looks really well preserved. But you can see this gentleman here, 18 years old, and you can see a very different pattern with a lot of fat infiltration in both the biceps and triceps. You can also appreciate, again, the variability between muscles. And even in Becker's, we see a lot of variability within muscles. So here, for example, we have the triceps of the arm. One part is well preserved, whereas another part is completely infiltrated. The same variability is observed in both the upper leg as well as the lower leg muscles. So overall, what we've seen is that there is significant fat infiltration. However, there is a lot of variability between participants, across muscles, as well as within muscles. And the pattern is less predictable than what we've seen in Duchenne. And I'll show you an example here. So here you see 2 gentlemen, and they almost have a complete opposite pattern. So here in this gentleman, you can see that the tibialis anterior is completely replaced by fat, whereas in the other gentleman, it's almost completely preserved. And the opposite is true when we look at the soleus muscle. So though on average, these 2 men have about the same amount of fat infiltration, the muscles that are affected are really different. So what we've learned from this, that in Becker's muscular dystrophy, we're going to have to develop what's called composite measures. They really take the fat fraction measurements of the multiple muscles and combine that into one measurement. And that's what we've been doing at this point. And we think this is going to be really valuable for clinical trials and make for a very sensitive measurement. What we've also been doing is looking at the relationship between fat fraction that we measure and performance on functional tasks. So how much does fat really influence function? For this purpose, we ask all of the participants to perform a battery of tests. This consists out of an assessment that's used typically for the arm, that's called the PUL. The North Star that was developed for limb-girdle muscular dystrophy, which is a motor performance test that can be performed in both ambulatory and non-ambulatory individuals, and then several walking tests, as well as a test to assess your ability to climb 4 stairs. At this point, we have taken advantage of these functional tests to really understand our data a little bit better. And specifically, Kelly Rock, who is a postdoc in our lab, has grouped the entire cohort of 52 individuals into 4 different subgroups based on their performance on the 10-meter walk-run, with A being the group that is the highest functioning and that can still run, and D being the individuals that are unable to walk. As is shown in the next slide here, you can see that overall, there is a really good relationship between fat fraction and performance on these types of ambulatory tests. So here, for example, you can see fat fraction in all of the muscles is really the lowest in the individuals that can still run. And as we see a decrease in the performance of the ability to walk, you can also see an increase in fat fraction across all of the muscles. We've also looked at the relationship between whole body fat fraction. So essentially it's composite measures that takes a combination of all of the fat fraction measures that we've done in these different muscles and compare that to performance on the North Star. And what you can see is a very strong relationship. So these data really confirm that MR measures of fat fraction are clinically meaningful in individuals with Becker's muscular dystrophy. We've also looked closely at the relationship between arm function, and this shows a really interesting pattern. So you can see when you look at the performance here on the PUL that we don't see a decline in arm function until about a fat fraction of 0.5 or 50%. So relatively speaking, the arm function is longer preserved. Finally, we're also looking at respiratory muscles in all of the participants. For this purpose, we perform pulmonary function tests. We take again these water fat images, but this time focused on the respiratory muscles. And we're also taking dynamic images using a cine MRI. So here the individuals are asked to breathe in and to breathe out, and we look at the changes here in the pulmonary cavity. So overall, we're very grateful. We've had a lot of individuals participating, and that has really allowed us to learn a lot about the disease progression in Becker's muscular dystrophy. And we are really getting a much better handle on developing these sensitive MR measurements that we think will ultimately be used in clinical trials. So at this point, I want to hand it over to Brian Springett, who is one of the study participants and who's also involved in today's event. And so we have a little video in which Brian introduces himself and also explains why he thinks it's important to participate in natural history studies.
A lot of those things that people take for granted, like sitting on the floor, walking upstairs, walking through grass, are all minor things for some people, but for a Becker patient, it gets hard. My name is Brian Springett, and I was diagnosed when I was 11 years old.
The goals of this study are to understand Becker muscular dystrophy better. And one of the unique things about our study is that once you come to our site and you participate, we will share that data with you.
One of the most important things is ensuring that the clinicians and the scientists that are working to find better care and treatments for our disease have the data they need. For me, understanding that was the most important part. Where else are they going to get it from other than the people that are living with those diseases? The other great thing about the study is that it's been super easy to be a part of. I at first was kind of nervous. I'm like, oh man, how much work is this going to be? How long is the day going to be? Am I going to be really tired? It was a 2-day process. One day being all about imaging and going through MRIs, and then the second day being the physical fitness testing. The team here has been amazing about communicating every last detail and then taking care of you, taking as many breaks as you need throughout the process. The biggest benefit to this study, not only for the scientists and clinicians to be able to receive the data, is for me to better understand my disease progression as well. In the end, if we can get more people to do this, we have more data for ourselves to use, and then hopefully we find better care for ourselves.
We can't do this alone. We cannot study the progression of this disorder without your help. You're the most important team member.